So as Sierra mentioned, today is all about HiChIP, how to get it into your lab and how to start working with the data on your computer. So a brief overview of what it is we're going to cover. We'll do a very, very brief intro into HiChIP and what it can do and why it's useful. Um, then we're going to pass it over to Miriam, our wet lab expert, to help you with some tips and tricks and some common questions that kind of come up during the workflow, just to get everybody aware of how do you generate these data. And then I'll pick back up and talk about data processing what you do with the data after it comes off the sequencer. So let's jump right in. So what is HiChIP and what can it do? So real briefly here's the workflow for ChIP-seq. You cross-link your cells, you fragment, you do your chromatin immunoprecipitation, and then you reverse your crosslinks, purify the DNA, and convert that into a sequence library, and you're done. Well, HiChIP follows a very similar workflow, except after the chromatin immunoprecipitation, you do proximity ligation, and then you reverse those crosslinks. And here now, if you're looking at the molecule that's generated from the assay, it's a chimeric read that has a biotinylated custom oligo bridge that sits right in the middle that captures that ligation event, and then you convert into a sequence library with a biotin pull down to enrich for your proximity ligated molecules, right? And so really what this does is it provides an additional layer to the protein DNA binding site. It allows us to view the interactome of that specific protein that you've pulled down against. So here, what we're showing is ChIP-seq, regular ChIP-seq with CTCF. And here you can see all of the enrichments for where that zinc finger protein binds to the DNA. And then here we're showing the HiChIP data. So we're showing the coverage as a one-dimensional plot, and then we're showing the interactions between each peak as an arc here. This is that interactome, right? So it really allows us to not only see where those, potential chromatin boundaries are, but now we can say, okay, these two are really interacting with each other, and so are these two. So this is likely going to be a TAD. Here's going to be an internal loop within that TAD, things like that. So with this interactome data, what it really allows you to do is to add meaning to your ChIP-seq peaks, right? So what ChIP-seq will tell you if you're doing CTCF is where those chromatin boundaries may exist, right? Not all CTCF are involved in chromatin boundaries. But if you add in that interactive data, you'll be able to classify the role of each CTCF site. You'll be able to actually assign where those chromatin boundaries are, and you'll be able to identify TAD and loop anchors that are actually involved in that chromatin folding. If you're doing something like H3K27 acetylation for active enhancers, or enhancer marks, or H3K4 trimethyl for active or poised promoters, if you're just doing ChIP-seq, you're really just going to be finding where those sites are, right? But what HiChIP can tell you is it will allow you to be able to classify very easily what a super enhancer is by looking at how many promoters it interacts with. It will help you find those correct enhancer promoter interactions and annotate the correct enhancer to the promoter that it's actually engaged with. Now, if you're looking at other transcription factors, right, if you're just doing ChIP-seq, you'll be able to locate where that protein is binding to the DNA, and you'll be able to find where its nearest neighbor promoter is. But that's about it. With HiChIP, you'll be able to determine exactly what the regions of highest interactions are, right? So maybe it's with a different promoter downstream, or maybe isn't interacting with the nearest neighbor promoter. And it will help you discover new regions outside of that transcription factor footprint to investigate. So, if we go back here, ChIP-seq gives us where that footprint is, but it doesn't necessarily tell us what it's interacting with outside of that footprint. If you bring in that 3D interaction, you could say, okay, this footprint actually interacts with something that's, 15 kb away. What is that? And it gives you an ability to discover new areas that are linked with that transcription factor. And so before I pass it off to Miriam, I think one of the questions that comes up quite a bit in our conversations is, How is what we do at Dovetail different than other approaches? Well, the big punchline here is that we don't use restriction enzymes to fragment the chromatin. We use MNase. And this actually leads to a more direct interrogation of the chromatin. So here's ChIP-seq, and here's the MNase-based HiChIP from Dovetail, and here's a restriction enzyme-based HiChIP dataset. And here I've plotted where the restriction motif is for that cut site. And as you can see here, there's actually a double stack cut site, so it enriches the peak for the restriction enzyme-based data away from where the actual peak center is, right? And even more extreme cases where you have restriction sites that actually just flank the binding site, you're actually asking the question when you use restriction enzymes, what is the nearest restriction enzyme site to my protein anchor, and what are the proximity ligation events associated with that restriction site? Here we're asking, what are the nucleosomes that are interacting, that are bound to that protein, and then what are those nucleosomes interacting with? And even for a more extreme case, where the restriction sites don't even occur anywhere near the peak. And so what you're seeing here is that all the reads are enriched around the peak, but you really don't capture the peak signal at all. Whereas here we can see the peak, and we can see all of the neighboring nucleosomes and how those internucleomes nucleosomes actually interact. Nucleosomes interact with the protein binding site. So with that, I will pass it off to Miriam to tell us how to make these data.
Well, hi everyone, and thank you for joining us. In this part of the presentation, I'd like to walk you through the HiChIP MNase workflow, so you have a better understanding of what is involved in running the assay in the lab. The steps of the workflow that I'll be discussing with you are straightforward. They can be achieved using standard molecular biology techniques, so there's no training needed prior to running the assay. The HiChIP MNase workflow can be completed in 2.5 days, and that is going from sample to a sequencing-ready library. It consists of 6 stages and a total of 18 steps. Day 1 is for cross-linking, digestion, cell lysis to extract the chromatin fragments, and incubation of the chromatin with the antibody of interest overnight. So that would be stage 1, 2, and the first part of stage 3. Day 2 is for immunoprecipitation and proximity ligation. So that would be the second part of stage 3 and stage 4. At the end of stage 4, you would have generated proximity ligated DNA that was associated with an immunoprecipitated protein of interest. This DNA is then converted into a sequencing library on day 3, which includes stage 5 and 6. There are several safe stops throughout the protocol, and there are also QC steps built into the workflow. Now, this is an overview of the workflow. In the next slides, I'll walk you through the workflow stage by stage, and I'll share with you our tips and best practices. Starting with stage 1. In stage 1, the chromatin is fixed in the nucleus, then digested with MNase. Following digestion, the cells are lysed to extract the chromatin fragments. For cross-linking, we use DSG and formaldehyde. So formaldehyde cross-links protein to DNA and proteins to proteins that are in close proximity. DSG has a longer spacer arm. It's a strong protein-to-protein cross-linker, and it cross-links proteins to proteins that are more distant. So when you use DSG in conjunction with formaldehyde, it increases the long-range information captured in the library. When cross-linking, we rotate the cells in a standard tube rotator so they don't settle. Now, the HiChIP MNase assay does not require sonication. It will only rely on the enzymatic fragmentation or the digestion with MNase. So it's important to achieve a good digestion in order to generate a high-quality library. For optimal digestion, we recommend to freeze the cells before cross-linking. The number of cells you'll start the assay with depends on your protein of interest, but that number will influence the amount of MNase you're using to optimally digest the chromatin. So to help with an accurate cell count and to reduce cell clumping, we recommend to use a low-speed spin and a swinging bucket rotor when you harvest the cells, And we recommend to count before you freeze the cells. So you would harvest the cells at low speed spin, wash with PBS, count, aliquot the number of cells you're going to start the assay with, spin down, discard the supernatant, and then freeze the cell pellet by placing it at -80. Then you can proceed with cross-linking and digestion. We provide sample preparation guidelines for 1 million, 5 million, and 10 million cells. Now moving on to stage 2. This stage is a QC step that is included early on in the protocol that will allow you to confirm that the chromatin is properly digested. It will also allow you to quantify the yield of clarified lysate or chromatin that you recovered from the cell sample. So how do you do that? To confirm that the chromatin is properly digested, you will take an aliquot from the total lysate you recovered after cell lysis at the end of stage 1. If you're going to proceed with the rest of the protocol on the same day, you can place this lysate in your ice bucket. Otherwise, we recommend to store this lysate at -80 for later. So this aliquot is essentially cross-linked chromatin. You will first start by reversing the cross-links, degrading the proteins with proteinase K, then purifying the DNA. You will use this purified DNA to run on an analysis instrument. You can use either tape station, bioanalyzer, or fragment analyzer. You'll also quantify this purified DNA with a qubit fluorometer that will enable you to determine how much lysate you recovered from your cell sample. Here is an example of a digestion profile analyzed with a tape station. That first peak represents mononucleosomes. Here are the dinucleosomes, and here are the trinucleosomes. The profile we're looking for has 40 to 70% mononucleosomes. In order to determine the percentage of mononucleosome, you're going to create a region that includes that first peak, which represents the mononucleosomes. So on a tape station, this first region is between 50 to 250 base pair. This is where you see the first peak. It may vary with different systems, but you're always looking at that first peak. When you create this region, a region table will be automatically generated, and it outputs percent of total. So how much of your DNA is within 50 to 250 base pair? This is the percentage of mononucleosome, which is in this case, 52.22%. Here is an example of a digestion profile analyzed with a fragment analyzer. Again, here are the mononucleosomes, dinucleosomes, trinucleosomes, and based on the smear analysis, the mononucleosomes account for 48.4%, meaning these two profiles pass the QC. Now, a few tips that I would like to share with you here is it's important to load that tape to spec. So avoid overloading the tape because that can result in a misrepresentation of the digestion profile. Also, we recommend for a specific system, the tape to use. For example, for tape station, we recommend to use the HSD5000 tape and not the D1000. And that's important because it gives you a good resolution for the size or the range that we're actually looking at for the digestion profile. Now, if your chromatin is not optimally digested, so if the chromatin is underdigested with less than 40% mononucleosomes, or your chromatin is overdigested with the mononucleosomes accounting for more than 70%, we provide guidelines for how to adjust this digestion. Now that you've determined that the chromatin is properly digested and you quantified the lysate, you can proceed to stage 3. You will start by incubating the chromatin or your clarified lysate with the antibody of interest overnight. Now, the question is how much antibody and how much chromatin to use, which is also a question that's asked in ChIP-seq. To address this question, we validated a list of antibodies. This list includes CTTF, histone marks, and transcription factors. So what we did is we tested antibodies from different suppliers to make sure that the antibody recognizes the epitope under the assay conditions. We also ran different titrations. So for each antibody, we provide guidelines for which supplier we recommend, and what is the amount of chromatin and the amount of antibody that we recommend to use for the HiChIP assay. I've highlighted CTCF, trimethyl H3K4, and acetyl H3K27 because we also offer a HiChIP library preparation with these antibodies in-house as a service, if, for example, you're interested in a pilot study. Now, after incubation, you're going to pull down on the antibody protein DNA complex with protein AG beads. Now that you have the chromatin fragments bound to the beads, you're going to run the proximity ligation reactions, starting with end polishing, where you're going to repair the ends of the chromatin fragments. You will then add a custom-made biotin-tagged bridge or adapter to those repaired ends. Then the fragments that were co-localized in the 3D space are going to be ligated to each other through the bridge. And here's where you're going to generate that proximity ligated chimeric DNA molecules. After ligation, you're going to reverse the crosslinks, degrade the proteins, and purify the DNA with a bead cleanup. For the intra-aggregate ligation and crosslink reversal reactions, they are incubated for 1 hour, but they can also be incubated overnight for convenience. Also, all of these reactions are carried out in a thermal mixer with shaking. I'm just showing you an example of a standard thermal mixer that we use in-house that has a heating and cooling function. So there isn't really any specialized equipment that you need to run the assay in-house or in your lab. So now that you've generated the DNA or the proximity ligated DNA, you're going to convert it into sequenceable molecules in the last 2 stages of the assay. Stage 5 is library preparation. As I mentioned, there is no sonication step prior to library preparation, so you will directly proceed to library preparation. These steps are standard for any NGS library prep. So you'll first repair the ends, add the adapter, and purify the adapter ligated DNA. Then before PCR amplification, we include a streptavidin bead pull down step, which will be pulling down and enriching specifically for the proximity ligated biotinylated DNA molecules before you amplify. So we run the amplification on the beads. And then following amplification, we size select the library with price select beads. And then we run this library on a fragment analyzer, bioanalyzer, or tape station to check that it has the expected size range. So we expect to see the library between 350 and 1,000 base pair. And we expect to see that laddering in the final library, which is reflective of the laddering we see with the MNase digestion. So this library is now ready for paired-end sequencing on any Illumina sequencing platform. One thing I would like to add is that the protocol as written is specific for the Dovetail library prep module and primer set that are offered as add-on modules that makes the protocol more streamlined. However, if you prefer to use a library preparation kit that you routinely use in the lab, please reach out to us and we can let you know if that kit is compatible with our assay and how you can integrate it in the workflow. So before deep sequencing, what we recommend to do is, now that you have the library, we recommend to sequence it to 20 million read pairs in order to run a QC analysis to assess the quality of the library. And we're going to be looking at the hiciness of the library, so the HiChIP properties or the proximity ligation enrichment. And we're going to look at the chip enrichment of the library. And now Corey will walk you through that QC process that we run post-sequencing. Corey, I'll pass it back to you.
All right, so you followed everything that Miriam just said. You now have some libraries and you just got them back off the sequencer and you want to know, how do they look? Are they good? Can you move forward? All of that fun stuff. So just kind of want to talk about the basic core workflow, right, that all the data are going to have to go through in order to start really asking some biological questions or even asking some QC questions, right? So it starts with putting the laser pointer on so my mouse doesn't jump around. Okay, so it starts with the FASTQ, which is the raw data that comes off the sequencer. Then we're going to align, and we align with BWA, and I'll talk about that a little bit more in depth in a few slides to get your alignment. And then you feed your alignment into a pairs sorter. So if you've never done Hi-C work before, this is something that's going to be a little new. But essentially what this tool does is it distills your alignment into a text file that gives you the read name, the chromosome of read 1, and just the midpoint of read 1, the mapping chromosome of read 2, and just the midpoint of that mapping fragment. It doesn't take into the account the length of the molecule. It just looks for the midpoint of that alignment. So it takes your big BAM file and just really condenses it down into an easy to use text file. That's all a pairs file really is. And then you feed this pairs file into a contact matrix generation tool, either Cool Tools or Juicer. We'll talk about that in a bit more. But I do want to say that we have some easy to follow guides that I'll point you to that really just follow, I should say, really nucleome standards for processing any proximity ligation data. So all of the tools like HiChIP Pro or HiChIPs, things like that, Homer, all of those are following this exact same workflow. They just have a whole pipeline built around it. And we found that, not using a restriction enzyme kind of breaks a lot of those assumptions. But we'll talk about that a little bit more towards the end. So let's get into the process a little bit more in depth here. So as Miriam said, there's two real questions you want to ask of your HiChIP library. What's the proximity ligation stats? In other words, how Hi-C-like is my library? Am I capturing long-range information, short-range information, all that fun stuff? Then the next question you want to ask is, did my chromatin immunoprecipitation work? In other words, how chippy is your data? So we're going to show you how we go about doing that. So first, I think we need to set one definition here, which is what is a valid read pair. If you're thinking about Hi-C, restriction enzyme-based Hi-C, or if you've never worked with Hi-C data before, this is a good refresher. So what I'm showing here is the workflow of proximity ligation with fixed chromatin, digest that chromatin, perform proximity ligation, and then convert that into a library for both restriction enzyme-based and DNase or MNase-based, right? Not using that restriction enzyme. So when you digest chromatin with a restriction enzyme, it cuts at these star sites for restriction sites. And then when you use something like MNase, it cuts randomly. And I've sort of made the color bleed over that fragment just to show that it's a continuous, it's not a hard stop cut site. Then you ligate. And when you ligate, based on proximity ligation, there are three possible ligation events that could occur. You could generate a self-ligation event where this molecule here comes in and makes a circle, right? The next is a religation event where you cut and then immediately ligate back to the original cut site. And then you have what's considered a valid ligation where these two ends here that are really far apart in linear space but are really close in three-dimensional space ligate back. And so the things to consider here is that if you're using a restriction enzyme, is that cut site, that the ligation event associated with that cut site occurs near a restriction site, right? You need to be within some distance away from a restriction site to really ensure that you are in fact capturing a ligation event. That's the first consideration. The second consideration is that the insert size or the distance being captured by that molecule is bigger than the sequencing molecule that you've generated, right? So let's take a step back and think about what that is. So Miriam just showed you in your library prep, right before you put it on the sequencer, you run it on a bioanalyzer tape station to look at the size distribution of your molecule fragments just to make sure they're small enough or the right size to go onto your Illumina sequencer. Right, and so your histogram looks something like this, maybe not so normal in distribution, but it fits within boundaries, right? So typically you're going to see between 200 and 350 base pairs at the lower end and between 800 and 1,000 base pairs at the upper end. And so the only way a molecule, a sequence molecule, has an insert size that's bigger than your library fragment, say your longest library fragment is 1,000 bases, and the insert size that's being spanned by read 1, read 2 is 20 kilobases. The only way that's physically possible is if a long-range ligation event took place and was captured in that biotinylation event. So that's something to keep in mind here, that it's really, when you're thinking about restriction sites, it's, am I close to it? And is that ligation event bigger than my molecule? Well, when you aren't using restriction sites, the only consideration is that your ligation event must be bigger than your library molecule. So a 1 kb cutoff there is actually pretty good. And then, one other thing that we want to point out here is our molecule size is so small after fragmentation that we don't have to shear. So what that means is these self-ligating events can't be converted into a library. So where here you would have these possible read types from the self-ligation in your restriction based noise, you do not get that noise signal at all in our data. There's no way to convert that into a library. If it's a circle, you can't ligate anywhere to that. So really the only data that are in the HiChIP libraries are religation events where it cuts and religates right back, or valid ligation events where the insert size is bigger than the library molecule. One last thing to point out here is that trans reads are considered valid pairs. It's a little bit, wishy-washy in the literature, but most matrix generation steps and most pairs parsers do not throw away trans reads. They retain them throughout the whole process. They might not be informative over cis regulatory elements, but they're still kept in the data. All right, so how do we go about doing this? Well, it starts with the alignment, as with any NGS data type. So here we take the raw FASTQs and we align with BWA with the -5SP. And what this essentially means is it turns a paired-end aligner into a single-end alignment, but it does this by using the pairs. So I'll walk you through it. So you start reading at the 5' end until you hit that either chimeric event or it starts reading the oligo bridge, and then it clips that alignment at the 5' end, and then goes and starts reading from the second pair at the 3' end until it sees the chimeric event or the oligo bridge, and then it cuts the alignment and then merges those two back together at the end. So it's a paired-end alignment that's done in a single end fashion. And this is actually the recommended aligner for any Hi-C data, restriction enzyme or not. The BWAMM with this -5SP flag is the sort of standard for dealing with proximity and ligation data. All right, so now we have the alignment, and there are really 3 categories that you could be in. The reads could not have mapped. The reads could have mapped, but with low quality. This is a Q of 40 here. Anything that's less than 40, we don't consider. And then mapped reads, which are anything greater than 40. From there, we feed this into pair tools, which will actually classify and remove PCR duplicates. So here we have those reads flagged here, leaving us with no dupe read pairs. And so in our little QC report, we have the total number of read pairs, and then the total counts for each of the categories of unmapped, mapped, PCR duplicated, and no dupe read pairs. And all of these are then reported as a proportion or percentage of the total read pairs. And here's just a graphical representation of the data where we're seeing about, a little over 80%, 82%, somewhere in there of mapping reads back. This is with the human genome. So that's kind of what we expect when we align any NGS data back to a genome. From there, we want to look at those no dupe read pairs and start asking how Hi-C-like are those. And we do this with pair tools. So first we bucket reads as either cis reads or trans reads. Cis meaning they occur on the same chromosome, trans meaning that they span the 2 chromosomes. And then from there, we want to look at the insert size of the cis reads, right? Because we already established that trans reads are valid and kept through the data filtering, so we don't throw those away. But we want to know if that ligation event is longer than the library molecule, so 1 kb. So we take these cis reads, again, this is a part of pair tools, that then basically just counts how many read pairs occur at less than 1 kb or greater than 1 kb. And then you could add some other cutoffs here that just basically say, okay, we're going to ask how many cis reads are greater than 10 kb. Whatever your definition of long range information is, you can definitely incorporate that into this question. Here we're just using 10 kb, and as you can see, here's the counts. And as the distance gets longer, that tail kind of forms. So we see that about 100,000 read pairs of this 2 million are cis less than 1 kb or invalid. Cis greater or equal than 1 kb are valid. So we have about a million, a little over a million. And of those, almost 900,000 are greater than 10 kb. And so now we can go through and bucket our reads as cis less than 1 kb as invalid, cis equal than or greater than 1 kb valid, and then trans reads are valid. And then we're showing this as a proportion of no-dupe read pairs. So here, cis is 71% of the no-dupe read pairs, trans is 28.72% of the no-dupe read pairs, right? And then here's just a graphical representation of that. All right, so now we know how HiC-like the library is. Let's start digging into the chromatin immunoprecipitation, or how chippy is the data. So first we start with ChIP-peak characterization. This is one-dimensional ChIP-seq data, not HiChIP data. So this signal could be coming from a ChIP-seq experiment where you've done max2 and identified your peaks. And really, we just do some very simple characterization here. We take the bed regions of those peak calls and we ask how many of them are there. And what is the mean and median length of all these peaks, and we report that as just total counts, and then as a size for both mean and median. And then from there, for other downstream analyses, we go in and we identify the center of each peak, and then we make a new bed file of these peak centers, which we will then use in the next step. So that's just giving us an understanding of what where those protein anchors, those protein DNA interactions actually occur, right? Just understanding how many of them there are, what's their size, and now we can start asking questions about how does the HiChIP enrichment compare to that ChIP-seq signal? Or how does that occur over the ChIPs, the enrichment regions, right? And so I'll walk you through this. So first we take those peak centers, right, from the ChIP-seq data, and then we build 3 new bed files, region files, of plus or minus 500 bases from that center, plus or minus 1 kb, plus or minus 2 kb for each one of those centers, right? And so as you go along the genome, we just make these windows bigger and bigger and bigger. And then we ask to capture the observed signal is basically what is the number of HiChIP no-dupe read pairs that are enriched in each one of these regions. So if we take this peak here, for example, we ask how many read pairs occur in this plus or minus 500 bases, how many read pairs occur in this plus or minus 1 kb, plus or minus 2 kb, and then we report that here as a count and then as a fraction of the no-dupe read pairs. Don't be super alarmed if there are data that, if it doesn't seem like it's that enriched, like 10, 12%. Keep in mind that only one read pair falls in that footprint anchor. The second read pair occurs someplace else in the genome as a function of proximity ligation. So we just actually want to know what that enrichment signal is for the observed signal. So now that we have the observed signal, the next thing we want to do is understand what the expected value is, so we have something to compare against. So what we're doing here is we are asking the question, what fraction of the genome is contained in these regions of interest? So plus or minus 500 bases, 1 kb, 2 kb, right? So if we count, we essentially ask how many ChIP-seq peak centers there are. That's kind of hard to say. And then we multiply that by the length of the ROI, that region of interest, right? So that gives us now, and then we divide it by the length of the genome, which gives us a fraction of the genome that's contained within these regions of interest. Then the assumption for the expected value that we're making here is that the no-dupe read pairs, all of the data, right, that made it through the mapping filter step, are evenly distributed across that region, which we know isn't true, but it's just to give us an expected value that, all the read pairs should fit within that fraction of the genome. And so that gives us a single value for single value for that fraction of the genome for coverage. And so now we have basically a flat line coverage value for all of the ROIs. So the next question we want to ask then is, what is the observed versus expected ratio? This is actually pretty easy. We know what the observed signal is because we've already calculated it, and now we know what the expected ratio is, or the expected value is, because we just calculated it in the previous step. And so now it's simply just dividing that observed signal by the expected signal, which gives us that enrichment. And so what we're really looking for here is that as we broaden the region of interest, as we go from plus or minus 500 bases to all the way out to 2 kilobases, that the reads accumulate a lower frequency as you get bigger, right? This means that all your reads are concentrated around that peak center. And so if you look over here, you'll see that as we make that region of interest bigger, that actual observed versus expected ratio goes down. That means it's strongest right within that 500 base plus or minus region of that peak center. So this is informing us that most of the data occurs right over the ChIP-seq peak center, not distributed widely across that whole footprint, which is what we want to see. And so that's very typical of any ChIP-seq experiment. As you broaden your window, you start accumulating less and less reads, and we just want to make sure that our data are following that same assumption. So the final thing we want to look at is, what is the aggregated enrichment signal across the whole genome. This kind of sounds complicated. It's really not. We're going to break it down for you. So what we do is we build a big table, and the columns in this table are the base positions from the ChIP-seq peak center. So minus 1 kb, minus 999 bases, minus 1 base at the center, plus 1 base all the way up to 1 kb. So every base position plus or minus 1 kb from that. And the rows in this table are each peak entry. So here's peak 1, here's peak 2, peak 3, and then your nth peak, right, goes at the very end of the table. And then what we simply do is ask how many read pairs cover that base for that chip, for that peak. So for the minus 1 kb base pair in this first peak, it may be 20, it may be 10 here, it may be 20 here, maybe 10 right here. And then as we get into the center, we're starting to look at, this is a bigger peak, so there's 2,000 base pairs that, or read pairs that cover it. There are only 1,000 base read pairs that cover this particular base at the center. And this is done with SAMTools MPileUp, embedded within a Python script the way that we do it. There's other ways to do it. This is just one way that we've shown here. And then, so now that we have this table that looks at how each base is being covered for every single chip position, now we want to understand what's that mean signal look like. So we take this whole table and we take the mean. So now we get a mean coverage score for every base at every single entry. And then what we want to ask is, what's the fold change from the mean for each position? So we then divide each entry in that table by the mean to get a fold change from the mean. And that gives us this resulting table here of fold change per base position for each peak. So then we take this table here, right? This is just step 3.2, step 3.2, picking up where we left off, where we have a table of fold change from the mean coverage for each entry. And now what we do is actually pretty simple. We just take the mean of each base position, or we just take the mean of every column. So now we have a position of minus 1 kb, and then here is the average fold change from the mean coverage. So the mean fold change from the mean coverage is essentially what we're asking. And then we take this here, and then we just plot it, where your x-axes are the base position and your y-axes are the fold change from the mean that we've calculated. So now that we know that our data are HiChIP, or sorry, are Hi-C-like and ChIP-seq-like, now we can go into contact matrix generation. And so before I jump into this in too much detail, I just want to say that there are a few ways to do this. The two main ones are with Juicer to build your .hic files, or with Cool Tools to build your cools or mcools. Normalization, Juicer builds in actually a bunch of normalizations into the contact matrix generation step. We typically find KR or coverage based normalizations tend to work pretty well, and Cooler just uses a balance function, which it isn't as flexible as Juicer's, but it actually does a fairly good job there as well. And so once you have your contact matrices, you are onto the analyses, and that is what our next webinar, or not next one, but the following webinar is going to be about. Is about really, how do you identify your one-dimensional peaks from your HiChIP experiment? How do you do significant interactions? And then how do you plot those, right? All that type of fun stuff. So we'll get into that more in depth at a later date. But I do want to point everybody real quick to this read the docs page. So I'm going to stop sharing real quick and bring up a different page here. And so what we've done is we've basically captured the second half of this webinar in this read the docs page. So it gives you a nice little overview of what HiChIP is, and then gives you a very in-depth tutorial of how to process HiChIP data. So it tells you what tools you need, where to go download them, what you need to do to your reference genome before you get started dealing with these data. Then we walk you through exactly how you do that alignment and pairs generation. If you're impatient, we have it all piped together here, so you could do it all in one step, or we break it out bit by bit, so you could just run the alignment, and then you can feed that alignment directly into pair tools and do the subsequent part of each of the pair tools filtering individually, so you get a sense of what you're actually doing to the data. And then we have a section on QC. We provide little Python scripts that help you generate these tables. And then we walk you through exactly how you would generate a contact matrix using Cooler or Juicer. So all of this is captured here on the Read the Docs page. We'll make sure that this link is incorporated into the presentation when we send it around to everybody. And so with that, I will stop sharing and say thank you very much.